Hello, let's talk about how things get through cell membranes. This is a shorter lecture, but it is incredibly important. So in one of our lectures, we talked about how the cell has this membrane, this plasma membrane around it. It keeps the inside of the cell separate from the outside of the cell, so you can get different things happening, and that's very important. But then how does the stuff on the outside of the cell go inside the cell, and vice versa? How is it if you have molecules that need to move into the cell or out of the cell, maybe hormones or sugars or whatever, how do they get through this plasma membrane? And there's several different ways, and this tends to be an idea that is incredibly important for anatomy and physiology, and in medicine and in biology and so many different things. But it's kind of definition heavy, and so it's very important to keep these things separate, but there's a couple different things that cells can do. So we're going to talk about something called unassisted transport. So these are two ways that things just move by themselves completely naturally. These stuff is doing this all the time, and you may see these words and like think, oh, osmosis, I know about reverse osmosis. That makes really fresh water. It's complicated. It's more, well, it's both more simple and more complicated than that when you're talking about anatomy. But that isn't all. So that's just stuff that happens naturally. Sometimes your cell needs to move stuff to where it doesn't technically want to go, and so then it needs some, well, it wants to go there, but it's too big to fit through the membrane, and so it needs some kind of carrier protein. So remember, proteins are a class of molecule made of amino acids, and they do so many different things. One thing they can do is just act as a bridge across that membrane to get stuff to move from the outside to the inside, or vice versa. It can do it two different ways. It can do what's called facilitated transport or active transport. We're going to talk about what this moving with or against the gradient means, but you'll notice anytime you're moving against the gradient, you require energy. So we're going to talk about that. And then there's bulk transport, where you're moving a whole bunch of stuff inside or outside of the cell. So if you have endocytosis, it means moving into the cell. So endo is into and cyte is cell. This requires energy. And phagocytosis is where you take in a whole lot of stuff. It also requires energy. And one more thing is called exocytosis. Is basically the opposite of endocytosis, where you have a vesicle that fuses with the cell and then pushes stuff out into the exterior environment. Let's go through these one at a time. So what is diffusion? So diffusion is for small and uncharged particles, or anything that is nonpolar or hydrophobic. So these are things that will cross the membrane because they interact with the lipids, but not with water. So they're moving against water, or anything polar. It's always from high to low solute concentration. And one way to think of this is if, well, let's look at some visuals. Let's say you drop, not that one. If you drop something, a drop of something into water. So we just dropped it. The molecules spread out randomly and they start bopping around. They bounce off of each other. They bounce off of the walls until they spread out to a pretty even distribution. Because they look like they kind of ball up a little bit, but they basically look like they're moving from this area where you put them originally in the really high concentration, and then they spread out as much as possible. So it's like getting on the bus. You don't want to sit right next to the one person that's there. That's weird unless you know them. So people try and spread out as much as possible when they get on the bus. Or if they go into a concert hall or something, people tend to spread out as much as possible. In the case of the molecules, they're doing it just by random action. It's called Brownian motion. But diffusion is just where you have a whole bunch of stuff in one place and then you spread out. So you're moving from a very high concentration to a low concentration, and that's called moving with the concentration gradient. So here's another, oh, you can't see it, hang on. Here's another picture. You drop in some dye into some water. It's going to spread out, so it's really high concentration here. There's no dye molecules here, even though there's water. The dye molecules will spread out until they're pretty evenly spaced. And that, again, is diffusion. So diffusion is important. It will just naturally happen. If you're diffusing into the cell membrane, that's only going to happen for really small, uncharged particles and things that are non-polar. It's going to happen, again, from high to low. So if you're moving high to low, you don't need any energy because it's just the natural thing. If you're a small particle and you're doing this, you don't need any membrane protein, and there's no energy required. So very important. The counterpoint to this is osmosis. So we see what happens if water wants to move between the membranes. So water can also move across the membrane because a cell membrane is considered semi-permeable. So it's only permeable, or it can only let things through that are a certain size or only certain things. And water does this naturally to compensate for a concentration gradient. And so it's well, let's look at another thing. I have a nice, there we go. So here are some sugar molecules. They're too big to fit across this membrane. So what does water do? Water is always going to spread out, so it is spread out as much as possible. Nope, it's going to spread out, it's going to spread out so it's got an equal amount. So water is all about having equal concentrations on either side of water, okay? So if you have no sugar over here, then you have a very high water content. If you have lots of sugar over here, you have a low water content. So water is going to spread out until the water content is equal. Okay, so this is actually moving from a high water concentration to a low water concentration. So it's the same idea that you're going from a high concentration to a low concentration. It's just weird in this case because it's specifically water. So this happens because of water pressure. We see this in so there's 3 different definitions we really need to know for this. And that is, where is it? Hypotonic, hypertonic, and isotonic solutions. Let's see if I can find a nice image of that real quick. So that is where if you have, let me see, sorry, I should have this up. If you take something and you put it into a fluid, here we go, this is a perfect picture. So let's say if you have an isotonic solution, that means that your cells or whatever you put in there have the exact same concentration as whatever's outside. So the same number of water molecules enter as that leave because they're not trying to rush to the point where it doesn't have any. They're not rushing against a concentration gradient because the concentration gradients are the same. On the other hand, if you have a hypotonic solution, that means that hypo means less. That means you have the outside solution has less concentration of other stuff, which means it has a lot of water. So if there's a lot of water out here, if you have like red blood cells, which is what these are, or if you have potatoes, or if you're a freshwater fish, the problem that a freshwater fish is always dealing with is that water is constantly entering its body because the water around it is very, very fresh. It's hypotonic, whereas living beings are full of different kinds of stuff. They're full of salts and proteins and so many things. So water wants to get in there and spread out as much as possible because it wants the inside water concentration to match the outside. So if a freshwater fish were dealing with this and it didn't have adaptations, it would just explode. It would keep filling with water and then explode, and that's what these cells do because they don't have a way to regulate it. That's why when you are getting blood transfusions or if you're getting fluids, they don't just pump pure water into you in an IV pump. They pump saline into you because if they pumped pure fresh water into you, your cells would explode, and that would be very bad. If you're worried about the fish, what they do is they just pee a lot. So freshwater fish will urinate constantly to deal with how much water they're taking in through their skin. On the other hand, if you have a hypertonic solution, that means that your outside solution is really salty. So if you do an IV drip with very, very, very salty water for some reason, not the regular saline, isotonic saline, the outside water, they're going to have much higher concentration of salt on the outside, which means a lower concentration of water than what's inside the cell. So the water, again, wants to go to where there's less of it. It wants to spread out as much as possible so you have an equal concentration of water inside and outside, which means your cells are going to shrink and they shrivel up. So in a hypertonic solution, which means lots of salt, you get shriveling. In a hypotonic, you get bursting. And it's in isotonic where they have equal concentrations, and you do get movement, but it's balanced, and so you don't get any bursting or any shriveling. So this is what a saltwater fish is always dealing with. The saltwater environment is a lot saltier than the inside of a fish, and so it tries to deal with this by, it drinks a lot of water constantly, it has very little urination, and it tries to balance its salt levels in a different way. So there's your fish lesson, but it works the same way in the cells. So the cells can't do that individually. They're used to a very specific kind of environment. So we have to be cognizant of this when we're taking fluids in. So not just when you're exercising and drinking. So a lot of times, you Gatorade and Powerade are very much about getting those electrolytes. You got to replace your electrolytes because you're sweating a lot, and so you're getting rid of salts and water. So you want to replace your salts and water to maintain an isotonic environment. If you sweat a whole lot and then you just drink water, let's say you do this to an excessive amount. If you do sweat, pee, and then take in just fresh water and no salts, you could get a hypotonic situation. It's not very easy to do, and it's not super common, but it is important to get in an isotonic. So keep your body regulated by maintaining isotonic concentrations when you are taking in foods and waters. That was too much detail there, but let's go back over here. Okay, so what's happening with osmotic pressure? So when we have this happening, so if we go back over to this, so you'll notice there's no protein here that's picking up the water molecules and moving them. There's actually no protein that we know of that does that. There are proteins that will open up to allow water to flow more easily, but you can't actually control where water is going within the body, between the cells. You can only control where water is going by controlling where the salts and sugars are going, so whatever else is going on there. So this happens without any energy. It happens naturally. And it's always, again, from high water concentration to low water concentration. So it's a specific kind of diffusion that applies only to water, because it's the water concentration is going to where there's less water. But it's a weird way to think of it, because usually we think in terms of whatever's dissolved into water. So it's very specific in osmosis that we're dealing with the water concentration, not the stuff dissolved in the water, which is called the solute, whatever it is. So that is tricky to keep separate, but it's very important. Okay, so here again, here's some more diagrams of those blood cells in an isotonic solution. So red blood cells look like this because they lose their nucleus to make more room, and it's also just a better configuration to have the most surface area. We're going to deal with it. It's important, but not today's lesson. And so if you have an isotonic solution, again, the inside salt concentration matches the outside, so then water, as it's moving in, it's also moving out at the same rate, and you maintain the cell's structural integrity. If you have a hypo or a dilute solution that your cell is in, water is constantly moving into the cell until it explodes. If it's very, very salty outside, that's a hypertonic solution. It's very concentrated, and water leaves the cell and shrinks. And, both of these are structurally bad for the cell. It'll rupture, bad things will happen. In terms of blood cells, that's not a big deal. We make a lot of blood cells. But if it's something like a neural cell, a neuron, that's a very big deal, because we don't make very many of them. There is theory out there that we don't make any new neurons after a certain age, so we want to protect them as much as possible. Okay, so that was osmosis and diffusion. Those things happen naturally. You just slide through the membrane, no problem. What if you can't slide through the membrane because you're a large molecule like glucose, or you're a charged molecule like a salt? So in that case, you have to have some sort of transmembrane protein. So you have some protein that sits across the plasma membrane. So this is a cell. So this is like, we're zoomed in on part of a cell. This is the membrane surrounding the cell. This is the interior of the cell. The nucleus is over here somewhere. And this is the outside of the cell. Let's say you've just eaten a whole lot of food. You have a lot of glucose in your bloodstream. You have to get that glucose out of your bloodstream by pushing it into the cell. Okay, and there's many reasons. We'll get into it. So in order to do that, you have a transmembrane protein, this carrier protein, that will scoop up some of that glucose and then let it go into the cell. So that is very specific. It's only going to carry the very specific things that need to move. So if this is a glucose carrier protein, it's not going to pick up anything else. It's not going to pick up any hormones. It's not going to pick up salt. It is a lock and key situation like we talked about with the enzymes, where it's only going to pick up what it is designed for. Okay, so you'll notice this is very high concentration. This is low concentration. So this is the direction it wants to move anyway. All the glucose wants to move into the cell, because it has less concentration. It's a big crowd surrounding a concert. It wants to get in. And this is the doorkeeper that makes sure only certain ones get in at a certain rate. And it actually lets them through the doorway, okay? So this is a strategy for big polar molecules like glucose, or charged particles like ions. Because it's moving with the gradient. So the gradient always goes from high to low. So since you're moving with that gradient, you do not require energy. Important. If you're moving with the gradient instead of against the gradient, you don't need energy. It's going to happen naturally. So what if you need to move against the gradient? So that's going to be, whereas this was facilitated diffusion, which means helping out diffusion, because the only reason why you have to help it out is because these particles are too big or they're too polar to pass through the membrane. If you have something that actually wants to move against the concentration gradient, so from a low to a high concentration gradient, like if you're trying to really pull in a whole lot of some molecule, like really pull in all the glucose from your bloodstream for whatever reason, you're going to do active transport. And so what that means is you have to have a different kind of carrier protein that has to use energy. Remember, when we're using energy, we take ATP and we pop off that one phosphate and grab its energy, and we make something happen. So in this case, let me move this because it's annoying. Did not work. All right. Okay, so in this case, we have the calcium molecule. It's not going to be able to pass through, or calcium ion, it's not going to be able to pass through the membrane because it's charged, and so it's polar, and these guys are non-polar on this side. They don't want to deal with anything polar. They don't let it through. And let's say there's a high calcium concentration on the outside, like you're depositing calcium in some sort of, anyway, it doesn't matter. So there's a lot of high calcium on the outside of the cell. This calcium wants to move into the cell so it can relieve that concentration gradient. But for some reason, you need to move that calcium outside the cell. Maybe you're building bones. And so what you have to do is you have this carrier protein. The calcium has to bind with a specific carrier protein. You then have to have energy release, which then causes the carrier protein to change shape, and then floop, push that calcium outside the cell into that higher concentration, which means this is an active process, and you have to have energy to make this happen. Another example is the sodium-potassium pump, which we will talk a lot about right now. So let's see. Let me see what I want to say. So inside the cell, there's so many things going on. They're mostly negative. So there's a lot of proteins that have a mostly negative charge. DNA has a mostly negative charge. So does RNA, etc., etc. So let's say you want to move sodium out of the cell, and you want to move potassium into the cell. And this is very important. We'll talk about it later. So in order to do that, you have this sodium potassium pump. Let's see if this is better. Nope. I don't have a good picture of it right here. Let's see if I can find a good one. Sodium potassium pump. There we go. All right. And so what it is, you're basically exchanging something. So you're saying, I want, not this one. Usually draw this on the board. Okay. So we have some sort of sodium. It wants to move. For whatever reason, you want to move sodium outside the cell. Like if you're a nerve cell, we'll get to why that's important. You want to move it outside of the cell, and there is very high sodium already outside the cell, so you know you're going to need energy to make this happen. So that happens. You have 3 sodiums, they get pushed outside the cell using ATP to power this reaction. Boom, it goes. Awesome. But then it's kind of like a 2-for-1. So when you do that, you've now opened up this carrier protein on the outside. You have a low potassium, so K is potassium. You have a low potassium concentration outside the cell, but you need potassium inside the cell for something. So while this carrier protein is open, it's going to grab 2 potassiums and use more energy to get that to then push the potassiums inside the cell. So it's basically a 2-for-1 example of that active transport, where you have sodium moving out. It doesn't want to because it's really high out here. And then you have potassium moving in. It's going again from low to high. It doesn't want to do that. So in both cases, you have to have energy be released. These are both examples of active transport. So a couple more active transport examples are endocytosis and exocytosis. So endo means inside. This is where molecules are going to end up inside a vesicle. So if this is the inside of the cell, this is stuff outside the cell that like the cell really wants to take in, and it wants to take in a whole bunch of them at once. So it creates what's called an invagination, which is a little indentation. All the stuff kind of clusters in that little area, and then it just kind of pinches off a little chunk of the cell membrane that creates a vesicle. Remember, a vesicle is just a little membrane-bound part of the cell that can then move on those tracks that we talked about with the actin, was it actin? I think it was a kinesin. The kinesin molecule would then strut down the tracks like a little awesome dude carrying that vesicle to wherever it needs to go. So this is for really big molecules, like really big macromolecules, like big proteins that have been made somewhere else, like protein hormones, that need to come in and do something on a different scale. This takes a lot of proteins. It takes a lot of energy. Even though it is, because it's moving from low to high concentration, so it's moving against that gradient, so you know it has to have a bunch of energy. And the opposite of this is when you have stuff that it was made in the cell, and then you want to release it into this extracellular environment, so you just get the exact opposite process. This could be neurotransmitters, this could be hormones. This could be waste that the cell just wants to get rid of somehow. And so it just goes up, it kind of starts to bond with the cell membrane, and then bloop, just pops into it, and then pops that stuff out, and then becomes a part of the cell membrane. Let's talk about phagocytosis. So phagocytosis is for really big stuff, like when your white blood cells, like phagocytes, this is one of our white blood cells, goes and reaches out and grabs bacteria and just destroys it. And this is, again, very active transport. It takes a lot of energy. You have to rearrange the cytoskeleton of the cell to support these fake feet, these pseudopodia, which then reach out and grab the bacteria. So let's see if I can make this work. Yeah. So here's an example of a white blood cell. It's found a bunch of bacteria, and you can see it started to create this weird, it looks funny because it's not in the same plane as the focus of your microscope, but you can see it's starting to take these things in, and the bacteria doesn't know any better. It's just not, whatever. The white blood cells, again, engulfing this, taking it in, and its whole point is to take in those bacteria that are hanging out in your body, destroy them so they can't do anything else. They can't hurt you at all. And that is phagocytosis. There's another cool example. Let's find that. And you can actually find white blood cells just chasing, white blood cells chasing bacteria. If you get the right image, you can actually watch white blood cells chase bacteria cells through your body. So here's a white blood cell, there's some red blood cells, here's bacteria. The white blood cell has figured out, hey, I need to eat that and get rid of it so it doesn't hurt anything. And then it just goes and goes until it finally can do phagocytosis and engulf that bacteria so it can't do any harm. So you see it can grow in any direction. Oh, there it goes. It's surrounded it. The bacteria can't escape anymore. You go, little dude. You go. Good job, white blood cell. So that was phagocytosis. I'll put these videos up too so you can watch it. All right. That's how I record. Okay. So that is a lot. But let's kind of review a little bit. So during digestion, the concentration of hydrogen ions in the stomach rises. What type of transport must be involved if, let's see, concentration of hydrogen ions in the stomach rises to many times that within the cells lining the stomach where hydrogen is produced. So what type of transport is involved? So we have cells, they create a whole bunch of hydrogen and then they push it out into the stomach. So what could that be? We have a small polar molecule. It is probably either facilitated diffusion or just plain diffusion. Okay, so I would guess facilitated just to be safe because it is a charged particle. Sometimes the really, really small molecules like hydrogen can just pass straight through the membrane, but to be safe, facilitated diffusion is a good answer. It is, yeah, okay. So certain types of white blood cells encounter bacteria. They engulf these bacteria. Like we said, this is called phagocytosis. If you are trying to, let's see, oh no. Okay, glucose is kept at a homeostatic level, which means a useful constant level in the blood plasma, but it's constantly consumed within the cells, so the cells are using it up. So if glucose is really large, how do you keep that constant supply of glucose? How do you suppose a constant supply of glucose crosses the membrane? So it's large, it goes from the high concentration in the blood to the low concentration in the cell. That is, again, facilitated diffusion. If it was moving against the gradient, it would be active diffusion or active transport because it would need energy to work. And that is, short but sweet, how stuff crosses the membrane. So make sure you go through all of them. Make sure you know phagocytosis, how it's different from endocytosis. So in endocytosis, you're just looking at the cell membrane blooping off little bubbles. But in phagocytosis, it's much more, the whole cell has to get involved in chasing down things, and you get this rearrangement of the whole cell. You have this active transport where you're moving against the concentration gradient, so from low to high, and you have a carrier protein, but it requires energy. You have another thing is facilitated diffusion, where you move from high to low, which is what everything wants to do to be in a more stable state, which means you don't need energy, you just need a doorkeeper basically to let you into the cell. Water moves through osmosis from high to low concentrations of water. Make sure you know the difference between these three words. And diffusion is just how stuff naturally moves. So diffusion is the movement of anything from a high to low concentration, just through random action. All right, good luck. Let me know if you have questions.